Hello physiology students, welcome to your lab on osmosis and diffusion. Now this lab corresponds to the chapter on cell and membrane transport in your lab notebook, but be aware that they're the same three activities. And the first of these activities looks at the diffusion of dyes through agar gel. And this experiment takes about an hour in lab to complete, but if you're doing it at home, it's going to take much longer. Because we're using larger molecular weight dyes, it'll probably take 15 minutes to set up, but 24 hours to process before you're able to read that experiment. Now the next two labs involve osmosis, and that is the diffusion of water through a semi-permeable membrane. Unfortunately, we cannot do these at home, so if you are at home, you're going to have to watch this presentation and collect the data as they're generated during the process of these two experiments. Okay, so let's start out with our experiment on diffusion. So what is diffusion? Well, diffusion is the movement of a substance from an area of high concentration to an area of low concentration. For example, what we have here is we have a beaker that we place some dye molecules into. Now initially they're more concentrated on the left-hand side of the beaker, but over time they spread out and reach an equilibrium. The reason they spread out is because the water molecules are always in motion, and so as those water molecules move through something called Brownian movement, they carry the dye molecules around with it until eventually we reach this equilibrium. So let's do an experiment that shows another example of diffusion. So what we have here is a simple beaker full of water, and what we have now is some dye molecules, some methylene blue molecules, and we're just going to introduce those into the top there. So once we place those in the top, you can see that they start to spread out. Initially they're more highly concentrated up in the top of the beaker, but over time they begin to diffuse and move throughout the beaker until they'll eventually reach equilibrium. Now, while we're on the subject of talking about diffusion, we need to talk about the 4 or 5 factors that affect diffusion rate. Okay, the first of these factors is the solvent temperature. And so the hotter the solvent, the hotter the water in this case, the more quickly the diffusion is going to happen. And that's because as we heat up a liquid or a gas, the molecules in that liquid or gas start moving around more and more frequently and moving faster and faster and faster. And so as they're moving faster, they're carrying the dye molecules along with them. Secondly, the other thing that affects diffusion rate is the size of the molecule we're trying to diffuse. So in general, small molecules diffuse very quickly, whereas larger molecules don't diffuse very quickly at all. So that's going to have bearing on our diffusion experiment involving the agar gels. The next thing that affects diffusion rate is the concentration gradient. Now initially, if I have a whole lot of something on one side of the container but none on the other side, we have a big disparity in the concentration of our solute. And so that's going to diffuse very, very quickly. On the other hand, if I have just a little bit of a difference between one side of a container and another and the concentration of a solute, that diffusion is going to go much more slowly. And finally, diffusion distance also matters as well. So the longer a solute has to go, the longer it's going to take. And so this has bearing, for example, if you're trying to marinate a cut of meat for some kind of dinner that you're trying to make. If you have a whole side of beef and you're trying to marinate that thing, it's going to take hours and probably days and days until that marinade reaches the inside of the meat. On the other hand, if you cut it up into smaller pieces, you increase the surface area and reduce the diffusion distance, so that meat will proportionally marinate a lot faster. Okay, the last thing that affects the diffusion rate is the solubility of our solute in our solvent. For example, right now we have methylene blue, and that diffuses very well. One, because it's a small molecule, but two, because it's very soluble in water. On the other hand, some molecules will be nonpolar, and they won't diffuse quite as quickly in water. So be aware that solubility has an issue as well. Okay, now we're going to go on and talk about osmosis. Now, osmosis is a special type of diffusion. It's the diffusion of water across a semi-permeable membrane. Now, water acts just like any other solute. It wants to go from an area where it's highly concentrated to an area where it's not highly concentrated. But here, the concentration of water is affected by the solute concentration. So if there's solute present, that's an area where we have proportionally less water, and that's where water wants to go. In other words, water will always go to the area of greater solute concentration. And to say that even more succinctly, solutes suck. That is, a lot of solute is going to suck water towards the area of greater solute concentration. So let's take a look at an example. So here we have this U-shaped tube that's filled with water, that's our solution, and then we have sugar molecules on both the left and right-hand side of the tube. We have more sugar on the right-hand side of the tube than we do on the left-hand side of the tube. And that tube is divided by a semi-permeable membrane, which I will tell you now will allow our water to cross, but it won't allow our sugar molecules to cross. Now, if sugar could cross, it would simply go from the right side of the tube to the left because it would be going from sugar's high concentration to sugar's low concentration. But again, we said that the membrane won't let sugar cross. So what does water want to do? Well, where is water more highly concentrated? Well, it's more highly concentrated over here on the left-hand side because there's proportionally less solute, so there's got to be more water. So water wants to go from left to right. It's easier actually to think about the solute here. Where is there more solute? Well, there's more solute on the right. What do solutes do? They suck. That's right, the solute is going to suck water towards it, and that's going to force the water from the left side of the tube to the right side of the tube. And this is simple osmosis. Okay, while we're on the topic of osmosis, we need to talk about tonicity. Now, tonicity is a comparison of the relative solute concentration between two solutions or between what's inside of a cell and what's outside of a cell. So take a look at the figure up here at the top. What we can see is we have a cell right here, and then we have outside of the cell right here. So intracellular and extracellular. In this case, we have equal solute concentrations on either side of the cell membrane. And so we would say that these two solutions here are isotonic. They have the same solute concentration. As a result, water will move from left to right, but it will move from right to left with the same speed. On the other hand, what we have here is something called a hypertonic solution. The outside of the cell has a lot more solute than the inside of the cell. And what do solutes do? They suck. So this hypertonic solution is going to draw water out of our cell and cause it to shrink or crenate. And finally, on the bottom here, we have what we call a hypotonic solution. The left side has a lot less solute than the inside of the cell does. And so the inside of the cell has more solute. And what do solutes do? That's right, they suck. So this cell is actually going to suck water from the surrounding solution into the cell, and that's going to cause the cell to swell and potentially even burst.